Thank you for viewing this recorded webcast. Unfortunately, the audio at the very beginning of this webcast is a little shaky, but bear with it, it gets better. Thanks so much.
So thank you for joining us today. It is now my pleasure to introduce our speaker, Jamie Freeman from Horizon Discovery.
Thank you very much, Leah, and thank you to everyone who has joined us for the webinar. I'm from Horizon Discovery based in the UK, and I'm going to be talking to you about our use of CRISPR screening technology in order to improve the CHO cell host for biomanufacturing. There you go. So for those of you who aren't familiar with Horizon, we're a genome engineering company. We were founded over 11 years ago through the generation of isogenic cell lines for use in R&D and drug development and drug discovery. But the two technologies that I'll be talking about will be use of RAAV, which is a historical technology for us, and which has clear IP and freedom to operate in bioproduction, and CRISPR, where we have access to the three major IP suites, including the Berkeley IP for bioproduction through ERS. One of the important things about what we do is we don't just do bioproduction as a company. We do all things that are related to cell line, to genome engineering and cell line engineering. And that obviously touches on an awful lot of different areas of interest from a drug discovery and development perspective. But obviously you're here to hear about the impact on using CRISPR engineering and RAAV engineering in biomanufacturing. So I think what's important in order to frame the work that we're doing is to give a whistle-stop tour of the existing landscape. So where we are at the moment, how Horizon is trying to change that, and how we're going to bring in genuinely disruptive technology in the coming months and years. Before Horizon came along, I started at Horizon around about 4.5 years ago, and the situation at that point was that there were existing cell lines that were available for biomanufacturing that were proven from a regulatory perspective, and that provided established mechanisms for generating clones at commercially viable and relevant levels. However, the feedback that we received from the industry was that people were frustrated at the restrictions that were imposed in order to access the best available system, which we believe and still believe at this point in time is the GS selection system. And those restrictions included obligations around media use, the restrictions on which CMOs you can use, the ability to modify cell lines both from a licensing perspective and from the perspective of not having the enough sequence information to be able to play with the cells, as well as the financial barriers around downstream costs, as well as upfront costs. And in fact, we've also provided the cell lines directly to some CMOs, so you can access the use of the cells by going directly to them if needed. We are happy to allow further cell line modifications, and we actively encourage it and would like to help you do that if it's something that you're interested in. And if you identify particular modifications that you want to introduce, but are concerned about how to do that in an industry relevant setting, then again we can help with that through the use of our RAAV technology, but it's not obligated. We believe that this leads to an industry-leading combination of performance aligned with value and licensing terms. With regards to performance, this is data across all of the evaluations where we have been provided with data. So as you can imagine, a lot of people aren't able to share some of the data they've generated, but this isn't cherry picked. This is the complete amount of data that we've generated, and so that means that it covers different antibodies, different antibody architectures, different vectors, different clients across different countries, and we're consistently seeing gram to gram and a half per liter stable clones leading to multi-gram per liter, gram to gram and a half liter stable pools leading to multi-gram per liter clones, which is exactly what you'd expect from a GS system. However, what is absolutely key to all of this, and why I said at the beginning that what I wanted to do was set the context, is it is still a GS selection system. So while it's very good at what it does, and while it's the best metabolic selection system that's available, at the end of the day, what it allows you guys to do is to identify in as rapid a way possible the best expressing clones from a stable pool. What it doesn't do is actually fundamentally change the way that a CHO cell is able to process biotherapeutics and protein. It doesn't change the biology of the cell and increase the capacity of the cell from a bioproduction perspective, and that's what we want to start changing. So we want to build on the GS platform. We consider it to be that and exactly that. It's a platform. It's a platform for you guys to use for your antibody expression, but it's also a platform for us to build on and improve upon. So there are different ways of doing that through purely increasing the amount of product that's generated, increasing the QP of the individual cells, or retaining the QP but increasing the VCD, the growth rate at certain stages, maybe extending the length of culture time and having an impact on product quality. There are a number of different attributes that we can look at. But in the first instance, we focus on titer as an aspect that we have heard from you guys particularly, that is an area that would benefit from improvement. So the way that we're going to do that is we use CRISPR from the R&D perspective. So obviously that's going to be the coming slides to extend on that. Where we will take CRISPR to perform screens and then to validate initial genetic targets which are having a significant impact on performance of the cell line. And then once we've validated that from an R&D perspective, we can go back with our AAV, which has a lower efficiency and a lower throughput, but a clear IP position to allow us to generate lines with freedom to operate in manufacturing. So if I give you a little bit of sort of history around why it's taken us to this point to be able to do the CRISPR screening in the CHO cells. So in order to have a successful CRISPR screen, you need to have three moving parts that are working in parallel. You need to have a good library. You need to have a good protocol in place in order to introduce that library to the cells, and you need to have an assay that can then read out on the library that you've put in to find out which of the genes are actually having an effect. And without any individual one of those three, you can't do a screen. So if you look back a couple of years, the sequence information available for the CHO was not of sufficient quality to allow us to generate a high fidelity CRISPR library against the CHO with any kind of confidence. By sequencing the genome, we've now overcome that hurdle, and through the knowledge and expertise that we've got through our Dharmacon end, we've now generated a CRISPR library, initially targeting 20% of the genome for proof of concept, with the full genome coming in a matter of weeks. And that's based both on the sequence and an optimized design algorithm. I spoke a little bit about the tiling array that led to the optimized algorithm in a webinar back in March. So if that is of interest, then that webinar is available both on the BPI website and the Horizon website, or you can get in touch with me if that's something that's of interest to you. So that has led us to a very high fidelity CRISPR library. We've optimized the screening protocol to allow us to actually put that library into the cells. And we've spent a lot of time developing high throughput assays that are compatible with the systems that we need. So we wanted to make them FACS compatible. We've generated a validated fluorescence-based assay which is compatible with the whole genome CRISPR screening. So as you can imagine, putting all of those pieces together has been a substantial amount of work and taken a while to get together. But we're now in this position where we can really move forward and focus our resource on doing that. So the first stage for that was to do a proof of concept screen looking at fitness of the CHO. So we didn't require the titer assay, that came later, but this was part of the optimizing the protocol. And once we do that screen, so regardless of what the phenotype is, be it fitness or titer or cell size or anything else that you want to assay, the process that we go through is broadly similar. And again, as a cell line engineering company, this is what we do. We've carried out dozens of whole genome screens, not just in CHO, but also in human cells as part of our drug discovery service, target validation service. So the ability to perform and analyze the screens is something which is really a core competency for us. So essentially, you need to generate the cell line, collect data from the FACS, analyze it using NGS, perform the QC in order to be confident that the screen is actually providing relevant data, and then finally do the hit identification, which is generated using a bioinformatics pipeline. So we followed this through on both the fitness screen and the titer screen, which I'll tell you a bit more about in the coming few slides. We are starting with just a standard CHO cell line, and essentially without any selective pressure, we want to grow them for a number of generations. If the knockout that's been introduced through the CRISPR guide reduces the cell fitness, for example, for essential genes, then that guide will disappear. It remains stable in the population if it doesn't affect cell growth at all, and if it improves it, then you get an enrichment. So that's where the bioinformatics comes in. And as a result of that screen, so you can see this is the data from 2 screens to show the reproducibility with an R squared of 0.96. You can see that around about 20% of the library have a negative impact on the cell fitness, of which the majority are known in humans, and we picked out nearly all of the strong essential genes that we know are required in humans. So this really validated the actual screening approach. So this assay showed that we can introduce a CRISPR library into the cells and identify a phenotype, a reproducible phenotype that we want to. What's also useful from it is we identified a number of genes that improve the fitness of the CHO cells. And so these might be genes that are of interest for us to develop into cell lines, which have particular parameters which are optimized for different process conditions, or even those that reach a higher VCD, for example, as long as we can retain that QP. And then, without skipping forward too far, we've also looked at screening in order to identify pure titer. So the way that this works is we take a monoclonal expressing cell line, and we have 2 different assays, which are both FACS compatible, and I'll tell you about both in the coming slides. And then that will lead to an enrichment in guides for those that either improve the fitness of the cells, will lead to an enrichment, but then that can be controlled for. And also, obviously, those that improve titer will improve fluorescence, and then you get an enrichment of the guides at the back end of that. What is also important for our approach is that we're not doing single pass screens. What we want to do is a screen on the cells, identify a number of genes that have an impact, take an engineered cell and then rescreen on the back of that, because as you modify the biology, as you change the metabolism of the cell, as you change the ability of the cell to transit products through the Golgi or to secrete products, then that means that you'll shift the bottleneck, you'll shift the cell biological interactions within the cells, and so rescreening will then identify those synergistic interactions, and we can start to build up a number of edits which all have synergistic, multiplicative impacts on the final phenotype, which in this case is titer. So this is so as I said, we had two FACS-compatible fluorescent assays which we've developed. One is for use with monoclonals and can be adapted to use of cells that are already expressing a monoclonal. This one that I'm going to describe here is independent of the type of product that you're expressing. So it requires re-engineering of a cell line, but it is also a platform assay that, as I say, can be done on a multitude of different architectures. So what we do is we generate the product of interest with subunit 11 of GFP, and then that is stably expressed within the cells. The cells have been pre-engineered to express the large subunit of GFP under a DOX-inducible promoter. So this is constantly being expressed under the normal promoter, but obviously doesn't have any expression because you've just got this GFP subunit 11. When you add when you induce expression of the DOX, you get the GFP 110, and then you get self-assembly of a functioning GFP, which can be temporarily controlled by the inducible promoter. So at the beginning of or when you want to start the experiment, you add DOX and then you measure fluorescence. And obviously, it required a lot of optimization of assay window and parameters in order to get the right level of transit. That then allows the fluorescence to reflect the level of expression of your product as it transits through the cell. This was shown by this graph here, where we have a range of different expression levels, which we knew that's being measured from a product basis, and then that was reflected in the corresponding range of fluorescence. So we know that the fluorescence is proportional to the expression level of the product. So once we want to use that assay, we then have two arms of the screen. So we have the control arm where there's no enrichment, and you purely grow the cells, the mixture of cells. And then you do screening, and then, as I say, what that does is that allows us to then control for other factors that the CRISPR targeting sequences have. The second arm is the treatment arm, whereby we FACS for high-producing cells, leading to an enriched population. We go through rounds of enrichment, and then that can then be analyzed by NGS, and the different populations are then compared to identify the hits. So with regards to pure logistics, we infect with CRISPR an MOI of 0.3, and the coming experiment was done with a 20% genome, against around about 4,300 genes, with 10 guides per gene and 500 cells per guide, means that we were screening in excess of 22 million cells. The output of this is the following graphs. So with no enrichment, you had around about 8% GFP expression, and after 5 rounds of enrichment, that moves up to 24% that were in the highly expressed box. What's absolutely key to this is the following graph, the following very simple graph. Because this is a measure of productivity. The FACS traces are a measure of fluorescence, but the fact that the pools that were generated, so the pools that come out of the enriched population, have a significantly higher productivity than the pools from the control arm, shows that not only are we increasing fluorescence, but that fluorescence is representing an increase in productivity. So this is a scatter plot of very similar data, or the same data essentially. So what you can see coming out here is the actual genes that we identified after the bioinformatics analysis. So you can see the majority of genes have no impact or a negative impact on titer, and then you have this tail here where you're starting to see a positive impact on fluorescence. Over time, you can see how these genes actually fall out. You have the non-essential genes and non-targeting control are in this middle band. The core genes, the essential genes, as you would expect, drop off over time. And then you have these enriched genes, and you can see that as you go through the different sorts, you get better and better separation of the different populations. And this is our hit population of genes. So once we've done that, we need to analyze the hits appropriately. So what we've done is a second CRISPR knockout screen, so a validation screen from a separate clone that's expressing the same molecule, and that actually confirmed the hits from the initial screen, so that gave us very strong confidence in those hits. We've performed a pathway analysis on the full data set, so pathway analysis isn't limited to the strongest p-values, the strongest hits. You can do pathway analysis across the spectrum of hits that come out and start to identify where are the commonalities that are having an impact, even if not the strongest impact, are having an impact on the phenotype that you're looking at. And again, this comes back to the fact that this screening approach, this bioinformatics approach, this analysis approach is something that we do as a company well beyond just the CHO sphere. So it's something that we've developed a lot of processes and expertise around. After doing this pathway analysis, the genes that what came through as the strongest hits were involved in ER Golgi transport, vesicle formation, and the unfolded protein response, all of which would be those kind of intuitive cell biological processes that you want to go after. So as all of these things are coming through, we're getting more and more confidence in the hits that we've identified. So once we've done this, the next stage is to validate the hits through the creation of individual knockout cell lines of the hits that have come through, and then we need to assess those for the impact on antibody titer and product quality in order to ensure that what we're finding is are genes that genuinely impact the ability of the CHO cell to manufacture that product. Then in the longer term, we want to identify synergistic interactions. So, for example, do we take a hit that was involved in the UPR and a hit that's involved in improving ER Golgi transport, combine those two, and would we expect a synergistic interaction, an improvement of having the co-engineered line. And as I mentioned before, we're also going to run a number of screens on the back of engineered cell lines. And then finally, once we've got all of the confidence and we know what our priority list is with the hits, then we're going to re-engineer that with our AAV in order to have an improved cell line. So as I mentioned, that particular assay was based on split GFP and therefore has cross-platform utility. We've also developed an assay looking at monoclonal. So this has the advantage that it is a cold capture-based approach. And so that looks at the fluorescence of an antibody that has at least reached the outside of the cell membrane. So it's a more accurate representation of the physiology of secretion of an antibody product. However, it is restricted to only being able to be used on standard mAbs. However, that's still a very powerful tool because obviously a lot of people are expressing standard MABs, which we saw at the poll at the beginning of the webinar. And again, so it's a similar approach. You need to go through rounds of enrichment. And again, you can see that you get a significant enrichment as you go through the rounds into this positive gate at the top end. But we still need to then take this to the next stage and follow the same path as we did with the split GFP approach in order to identify novel targets. So that's what we're going to do. We need to perform the screen on an antibody expressing line, and then again go through the same process of validation, test to make sure that it has broad platform applicability. There's no point in optimizing a cell line for the expression of a particular monoclonal. This needs to be something which is broadly applicable across the industry. Look at different product quality attributes, identify the targets, re-engineer and release those lines. So I think what's worth noting as well is this particular assay is something which we can use in order to have this broad utility cell line to replace and refine and improve on the GS platform line. The split GFP approach is one that can be a little bit more bespoke. It was designed in conjunction with one of our pharma partners in order to specifically identify genes that improve the expression of difficult to manufacture products. So we can take in difficult to manufacture products, engineer the cell line slightly, as I mentioned, and then find targets which are specific to that particular product. Over time, as we iterate through this, we'll be able to identify common targets which work for different families of difficult to express targets. Which is something which can then be released on more of a generic basis. But in the short to medium term, I think that approach is going to be more restricted to individual products that people need to find solutions for the expression for. Whereas the approach for the monoclonals is one which can be, as I say, have broad utility to generate a new and improved platform expression system. So to summarize, we are a fast-growing provider of manufacturing cells to industry, and that's a consequence of having a good cell line with flexible licensing terms, good support, and accessible costs. While we are very happy with the cell line and our customers are happy with the cell line, we are focused on improving it substantially and really changing what the CHO cell is capable of in a bioproduction capacity. And our ability from a genome engineering perspective actually puts us in a relatively unique position to be able to achieve this. And finally, as I mentioned, we can improve your cells either through directed engineering of targets that you've identified, which we can then engineer using our AAV to provide freedom to operate, or we can take your difficult to manufacture products and actually do whole genome screening to identify specific genetic targets that improve the expression of that particular product family. And on that note, I'd just like to thank you for your attention and encourage you to ask me anything that you want.
Okay,
ideally related to the talk.
So we do have one question, and if anyone else has a question, go ahead and type them in. We are a little over time, so I'm going to try to get to a couple questions, and then you can follow up directly with Jamie, and we'll pass along any questions you type in the chat window to him as well, even if I don't ask them. So the first question is, are there any growth mediums and or supplements which work best with your cell line?
So we have recommended protocols. The cells were originally adapted into suspension growth in OptiCho from Invitrogen, LifeTech, Thermo. and we currently recommend the use of CHO for standard culturing. We've also partnered with a media development company in order to develop a media formulation which is specific to our cells, but that's a fair few months out from release at this point in time. So as I say, we provide recommended protocols, but there is an element of process optimization which is still left to be done. And then with regards to supplements, so the main supplements we have are around boosting the single cell dilution capability of the cells, or efficiency of the cells, I beg your pardon. And that we have done, so we've partnered with the Centre for Process Innovation within the UK to access some of their AMBER technology. So we do have feed strategies in place. But again, those protocols will get shared with you, and we have we take the view that we want to support you guys through the use of the cells as much as possible. So we have support available to work with you to make sure that you get the best use from the cells.
Okay, so this is the last question. Can you share the name or the publication of the CRISPR design algorithm that you discussed?
It hasn't been published, so that's sort of proprietary, internal to Horizon, just at this point in time. So
okay.
Unfortunately not.
So thank you, Jamie.
You're very welcome.
And thanks to our audience for joining us and bearing with us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll get an email providing with a direct link. We look forward to having you join us in the future. Look for the announcements in your inbox. Bye.
Bye.